So key topics today are going to be the electron transport chain in the mitochondria, capture of light energy in photosynthesis, which results in building up of the proton motive force. That's key. That's the similarity between the two. And then you get synthesis of ATP in the mitochondria and chloroplast from this proton motive force. Okay? So energy from reduced fuels is used to synthesize ATP in animals because we consume chemical energy and we make our ATP from reduced fuels. These electrons are transferred to reduced cofactors, right? We've already seen how we produce a lot of NADH or FADH2, whether we're burning sugar, that gets sent to the TCA cycle. We produce some in glycolysis. The TCA cycle produces more. These electrons have to go somewhere, and we capture the energy from transferring those electrons in the electron transport chain. In oxidative phosphorylation, the energy of NADH and FADH2 are used to make the ATP, and that's captured by this electron transport chain. We're somewhere. Oh, in the mitochondria, I guess. So we're going to capture the energy, and what we end up doing is building like a proton gradient across a membrane, and that energy is stored in that proton gradient. As those protons flow down their electrochemical gradient, that energy is used to produce ATP from ADP and free phosphate. So the energy flow in cellular respiration, again, glycolysis, we capture electrons in the form of NADH in glycolysis. Same with fatty acid oxidation. We're going to capture electrons as we strip off each one of those acetyl-CoAs. Then when we get to the acetyl-CoAs, the citric acid cycle yields more electrons in the form of NADH or FADH2. And these NADHs and FADH2s, the reduced electron carriers, then transfer their electrons via the electron transport chain, and that energy, ultimately the electrons end up with oxygen. That's the ultimate electron acceptor to form water. We reduce the oxygen to the water, and that energy is used to produce ATP. And this is called oxidative phosphorylation because we phosphorylate ADP to ATP, and we use oxygen in the process. Electrons from the reduced cofactors NADH and FADH2 are passed to proteins in the respiratory chain. In eukaryotes, oxygen is the ultimate electron acceptor for these electrons, and the energy of oxygen is used to phosphorylate ADP. Things I just said. Same pictures over and over again, basically. Plants, however, they do something a little bit different. It's called photophosphorylation rather than oxidative phosphorylation. So the key differences with oxidative and photophosphorylation is where do you get the energy from? Oxidative phosphorylation, we get the energy from reduced biofuels like fats or carbohydrates. Plants get the energy from light. So light causes a charge separation between chlorophyll molecules. So you boost electrons. Those electrons can then be transferred to a cofactor, NADP, to produce NADPH, and it reduces NAD. So the energy of the oxidized and reduced chlorophyll molecules then used to drive the synthesis of ATP. Water is the source of electrons. So rather than with oxidative phosphorylations, oxygen is the ultimate electron acceptor. In plants' case, they use water as the source of electrons. So you take reduced oxygen, you strip the electrons from that reduced oxygen to oxidize the water and produce oxygen as the byproduct. Does that make sense? So it's kind of in opposite. So photophosphorylation, you get energy from the sun. It's going to boost electrons in the chlorophyll. the electrons ultimately come from water, and you produce oxygen as a byproduct. These electrons transfer to NADP to make NADPH, and if you remember, NADPH is the cofactor that's used for anabolic processes where it gets electrons for that. So this will be used ultimately to make sugar and lipids and other things along with the ATP. And it's this chemiosmotic theory is how we get the ATP. So taking ADP and free phosphate and forming ATP is really thermodynamically unfavorable, right? You have a lot of negative charge with ADP, and then we're going to add another phosphate to that, add even more negative charge, so you have all this negative charge bulked up together. You lose the resonance from the phosphate when you add that to it. So it's really unfavorable. So how does this happen? Phosphorylation of ADP is not the result of a direct reaction between the ADP and some high energy phosphate carrier. That was back in the 50s and 60s. They were sure there was some high energy electron carrier that was able to do this. Okay. And I don't know if they say the guy's name, I forget his name, but this one guy proposed this chemiosmotic theory, and everybody made fun of him and ridiculed him, but then ultimately he won the Nobel Prize because it turned out correct that there's this chemiosmotic theory. So the energy needed to phosphorylate ADP is provided by the flow of protons down its electrochemical gradient. And the energy released by electron transport is used to form this gradient. So we have the reduced cofactors. They have electrons. They're going to transfer those electrons to proteins in the gradient. And you take a small stepwise going down those electrochemical potentials. And at each step, you can pump protons across the membrane against its gradient. And it captures a little bit of that energy from that electron being transferred down its reduction potential, basically. So when you produce that gradient, that's the energy stored from those electrons. As those flow down the electrochemical gradient through the ATP synthetase, it causes conformational changes that can force ADP and phosphate to form ATP. And we'll see how that works. So the chemiosmotic energy coupling requires membranes, right? So we have to be able to separate those charges and not allow or control the flow down their electrochemical gradient. And the proton gradient needed to form ATP can be stably established across a membrane that's impermeable to ions. This includes the plasma membrane in bacteria, inner membrane in the mitochondria, and the thalcaloid membrane in chloroplast. So we share the same type of process to form ATP. Membrane must contain proteins that will couple the downhill flow of electrons in the electron transport chain with the uphill flow of protons. So these electrons flow down their reduction potentials, and at the same time, the energy as they go down can be used to pump protons against their electrochemical gradient to form a pH difference across those membranes. Membranes must contain a protein that couples the downhill flow of protons to the phosphorylation of ADP, and this is the ATP synthetase. And this is how it looks in the chemiosmotic theory. It looks in mitochondria versus chloroplast, right? So this is the inner mitochondrial membrane. This is your ATP synthase and your electron transport carriers. Electrons get transferred from NADH or FADH2, and they go down. They get transferred from one subunit to the next, and at each step, you're able to pump protons against their gradient, so you generate a high concentration of protons, which would equal a low pH. And these protons want to move down their electrochemical gradient, and the only place they can do that in the membrane is this protein, the ATP synthetase. And as they flow down, they're able to force ADP and phosphate to produce ATP. come together. The energy from this gradient causes that. The same kind of thing happens in the chloroplast, but instead of having the reduced electron donor, we use light to excite those electrons, and it splits. Those electrons come from water, and then they flow down their reduction potentials the same way in the electron transport chain, and you produce a proton gradient on the inside of that chloroplast across the alkaloid membrane. And then the protons flow down their electrochemical gradient again, producing ATP from ADP and phosphate as you get that shift in conformations. So the structure of the mitochondria. So you have a double membrane which leads to 4 distinct compartments. You have the outer membrane, it's relatively porous, allows passage of metabolites in and out of the mitochondria. The inner membrane space has a similar environment to the cytosol, except you have a higher proton concentration or a lower pH. And this is where you generate that proton gradient. You pump the protons into this inner membrane space. And then your inner membrane is where you get that high potential of protons across from the inner membrane space to the matrix. It's relatively impermeable with the proton gradient across it. Location of electron transport chain complexes are on this inner membrane. Convolutions called the cristae serve to increase that surface area. If you've ever seen pictures of the mitochondria, there's one right here. You have lots of convolutions, and that increases the surface area, so you can have a lot of surface area to produce ATP and transport electrons. And then the matrix is where we do the citric acid cycle and parts of lipid and amino acid metabolism. Lower proton concentration, which equals a higher pH, right? Negative log proton concentration. And then here's your picture again showing you the structure of the mitochondria. So the electron transport chain complexes contain a series of electron carriers. And each complex contains multiple redox centers where the electrons will go from one center to the next to the next. And they contain flavin mononucleotide or flavin adenine dinucleotide, which has the same, active part of the molecule here. The initial electron acceptors for complex 1 and complex 2 can carry 2 electrons by transferring 1 at a time. You also see cytochromes A, B, and C, and there will be a lot of iron-sulfur clusters that will accept these electrons. Cytochromes are 1 electron carriers, right? Because you're just shifting between oxidation states of iron. So when you have iron protoporphyrin 9, heme C, and then heme A, and A-type cytochromes. This is C-type cytochromes, and this is what we see in B-type cytochromes. So they're only going to be able to accept 1 electron at a time, transfer 1 electron at a time, right? Because you can go between Fe plus 2 and Fe plus 3. More 1 electron carriers include these iron-sulfur clusters. And they're coordinated by cysteines in the protein. You have the iron. And again, it's iron, so it's only going to be able to switch between plus 2 and plus 3. So you get transfer of 1 electrons between these, iron-sulfur clusters. Another electron carrier in the electron transport chain are called coenzyme Q, or ubiquinone. And it's a lipid-soluble conjugated dicarbonyl compound that readily accepts electrons. This is a 2-electron acceptor. It can transfer 1 or 2 electrons. So upon accepting 2 electrons, it picks up 2 protons to give it to form an alcohol, which is ubiquinol. Then ubiquinol can freely diffuse in the membrane because it's a large lipid-soluble compound. It diffuses between complexes in the electron transport chain. It's a mobile electron carrier transporting electrons from complexes 1, 2, and 3. And we'll see how that works. And this is the structure of its active site. Well, here's their whole structure here. Long aliphatic chain. And this is the active site. You can go between ubiquinone, the quinone form, semiquinone radical, where if it only has one electron accepted or donated, or when it takes 2 electrons, you form the ubiquinol, the fully reduced alcoholic form. And we'll see cases where it goes from this to that in the electron transport. And these are a table showing your standard reduction potential, how it flows. So you ultimately, we start off with NADH, and then we work our way down. Water is the ultimate electron, or not water. Oxygen's the ultimate electron acceptor in the electron transport chain, and that's where it ultimately ends up. So we already saw this, right? Reduction potential, where we can calculate the change. And reduction potential is gonna, for negative free energy change, you need a positive change in reduction potential. And electrons are transferred from the lower, more negative, to the higher, more positive reduction potential. Free energy is released, is used to pump protons, storing that energy. And we just do it a little bit at a time. If we went straight from our NADH to oxygen, that would just release a lot of heat. And we do see that. Where do we see that? Or the FADH2 to oxygen. Do you guys remember? The peroxisomes. You can still burn lipids in the peroxisomes, but then the reduced cofactors just go straight, the electrons go straight to oxygen. So you don't get the ATP formation out of it, but you do generate heat. The glyoxysomes will do the same thing. And this is where we get the flow of electrons from biological fuels into the electron transport chain. So you have complex 1, complex 2. So complex 1 will accept the electrons from NADH that we form in the TCA cycle or glycolysis. And it goes from FMN, which is a flavin mononucleotide, to an iron sulfur center, to ubiquinone. And then the ubiquinone is a mobile carrier, and it'll transfer those electrons to complex 3. Complex 2, and in this process, we pump protons across to the inner membrane space. Complex 2, however, takes those electrons from FADH2. And the example directly in the TCA cycle is going from succinate to fumarate. Those electrons go from FAD to the iron sulfur center and then to ubiquinone to go to complex 3 eventually. So, and theoretically, complex 2 does not pump protons across the membrane, so you get more energy from the electrons coming from NADH than you would from FAD. And I like this figure, it's from the old book, but it shows you the different parts of each complex and how the electrons will flow down their electrochemical, not the electrochemical, the reduction potentials. This is reduction, reduction potentials for the different parts of complex 1, complex 2, complex 3, and then complex 4 in the electron transport. So at each step as they go down, you can pump protons across the membrane and generate a gradient. And you see complex 2 is about equal with complex 3, so you don't get any pumping of protons when you go through complex 2. So complex 1 is also known as the NADH ubiquinone oxioreductase. One of the largest macromolecule assemblies in the mammalian cell. over 40 different polypeptide chains encoded by both nuclear and mitochondrial genes. NADH binding site is on the matrix side. Non-covalently bound flavin mononucleotide accepts those 2 electrons from the NADH. And then you have several iron sulfur centers pass 1 electron at a time toward the ubiquinone binding site. So the NADH, boy, that arrow's backwards, isn't it? Huh? It should be going from NADH forms NAD plus hydrogen ions, right? Those 2 electrons go to FMN, which then get transferred to a series of iron sulfur centers. And then, that's 1 electron at a time through the iron sulfur centers. Then to ubiquinone, and in that all these electron transfers, we're able to pump 4 hydrogen ions across the membrane against their gradient. So NADH ubiquinone oxyreductase is a proton pump. Transfer of 2 electrons from NADH to ubiquinone is accompanied by the transfer of protons from the matrix to the inner membrane space. Experiments suggest that about 4 protons are transferred per 1 NADH. And reduced coenzyme Q then picks up 2 protons also. Because you go from the quinone form to the alcohol form, you need those 2 protons there also. Protons are transported by proton wires, which is a series of amino acids that undergo protonation and deprotonation to get a net transfer of proton from one side of the membrane to the other. So normally you think of wires as transferring electrons. These are amino acids that can accept or donate protons, and they just flow down the protein against, to get transferred across the membranes. Complex 2 is, we've heard this before, succinate dehydrogenase, right? So we remove a hydrogen from succinate, and that hydrogen comes with electrons, and we reduce FAD to FADH2. And that produces fumarate, which can then go on through the electron transport, or TCA cycle. But these electrons then go through the electron transport. So FAD accepts the 2 electrons from succinate. Electrons are passed 1 at a time, again via iron sulfur centers to ubiquinone, where it becomes reduced, ubiquinone, or ubiquinol, because it's the alcohol, right? And does not transport protons. So that's the difference. Complex 2 does not transport protons. We get protons transported with complex 1. And this is the figure showing that. So we have FAD here that accept the electrons. They go through multiple iron sulfur centers, and ultimately to heme B and C, and then to ubiquinone, right? Ubiquinone then can diffuse into the membrane as ubiquinol, and ubiquinol can diffuse laterally through that inner membrane and find its way to complex 3. And complex 3 is known as ubiquinone cytochrome C oxyreductase. Uses 2 electrons from ubiquinol to reduce 2 molecules of cytochrome C. So 2 electrons from 1 molecule of ubiquinol, because remember ubiquinol is a 2 electron carrier. Cytochrome C, it's a cytochrome, so it only has 1 iron there. It can only accept 1 iron at a time. So with 1 molecule of ubiquinol, you're able to reduce 2 molecules of cytochrome C. Okay. Additionally, contains iron sulfur clusters, cytochrome Bs and cytochrome Cs. The Q cycle results in 4 additional protons being transported to the inner membrane space. And this Q cycle you got to look at it. It's real kind of convoluted process, but we'll see. This is where we see our semiquinones come out in the electron transport. So this is complex 3. And there's a number of different sites. You have your heme Bl, heme Bh, which are electron, single electron carriers. You got 2 iron sulfur centers at the QP site, the heme C1 site, and then cytochrome C itself is one of the other mobile electron carriers. However, it's water soluble, so it carries the electron from complex 3 to complex 4 via the intermembrane space. So this is not lipid soluble, but it's a water soluble carrier, unlike ubiquinol. And we'll look at this Q cycle here. So experimentally, 4 protons are transported across the membrane per 2 electrons that reach cytochrome C. 2 of the 4 protons come from ubiquinol. The Q cycle provides a good model that explains how 2 additional protons are picked up from the matrix. 2 molecules of ubiquinol become oxidized and release those protons into the intermembrane space. 1 molecule becomes re-reduced. So we oxidize 2 and then we re-reduce 1. And thus a net transfer of 4 protons per reduced coenzyme Q. There's 2 stages to this Q cycle. So then it's kind of convoluted. So you have your ubiquinol out in the membrane. It'll diffuse to the complex 3 and bind. And then it gets split. 1 electron goes to this iron sulfur center, then to cytochrome C1 or heme C1, and then to cytochrome C. And in that process, we pump the 2 protons out, and you have ubiquinone again, and that comes out back into the membrane. The other electron gets transported down, through other electron carriers to this site where you have ubiquinone, and you form the semiquinone, and then it goes back to this site as the semiquinone, getting prepared for stage 2 of the Q cycle. You have the semiquinone, another ubiquinol comes in, those electrons are split again, and again, 1 electron ends up at another cytochrome C, pumping 2 more protons across. The other electron goes down and re-reduces the semiquinone that was formed in the first stage to produce ubiquinol again, which will go back into the membrane and be able to diffuse back to here again. So the net equation is with 1 ubiquinol, 2 cytochrome C1s become oxidized, or they are oxidized. 2 protons, your net result is you have oxidized ubiquinone, 2 cytochrome Cs that are reduced, and then 4 protons that move against their, into the intermembrane space, the P side. And this is the Q cycle, cycle 1. And like I said before, the reduced ubiquinol binds, the electrons get split apart to an iron sulfur center, to heme C1, and then to cytochrome C in the intermembrane space, pumping 2 hydrogen ions across that into the intermembrane space. The other electron goes down to form a semiquinone form of ubiquinone, because it accepts 1 electron, and then goes back to this cytochrome B site. And that's the first cycle. The second cycle, then again, another ubiquinol comes in. 2 more hydrogens get pumped. The 1 electron goes back to cytochrome C. The other comes back and forms ubiquinol again from the semiquinone. And then this ubiquinol can then be available to go into this site again. And I just put a number of different figures from different books showing you the same thing. This is the old book. I don't know if it helps explain it better, but I tried to include more than one way because just in case you get it one way better from than the other. So. And this one has the sites labeled as the QP site that accepts the ubiquinol, and the QN site also. And then this is again the second site. And then there's a little animation that you have to watch really quickly to see what's going on. Because it kind of moves fast. So Q cycle, is it moving stage 1? It's hard for me to... Do you see that? And you see it has the little half arrows for single electrons going places. And then shows you where the ubiquinol will replace that. 2 protons go across. It's hard to describe this as it's doing it. We could slow it down, perhaps. So we mentioned that these electrons end up in cytochrome C, and that's the second mobile electron carrier in the electron transport chain. There's 2 mobile electron carriers. This is a single electron carrier versus the 2 electron carrier, which is... Do you guys remember? We were just talking about it.
Ubiquinone?
Yeah, ubiquinone or coenzyme Q. Any one of those words, ubiquinol. The second mobile electron carrier is the heme-containing protein, cytochrome C. Heme iron can be either ferrous, the oxidized form, or the ferric. So it's only going to be able to accept 1 electron. So it carries a single electron from cytochrome BC1 complex to the cytochrome oxidase enzyme, which is complex 4. And cytochrome C will absorb visible light. Intense sort band near 400 nanometers absorbs blue light and gives the cytochrome C an intense red color. Similar to like our hemoglobin gives that red color. You know, it's iron. Cytochromes are named by the position of their longest wavelength, the alpha peak, which is right here, right? So they get, that will vary based on which cytochrome it is, and that's how they get their name, cytochrome A, B, and C. Cytochrome oxidase is complex 4. So what do you think that's going to do? That's going to oxidize cytochrome C. So mammalian cytochrome oxidase is in the membrane protein with 13 subunits. It contains 2 heme groups, A and A3. Contains copper ions, so we need copper for this electron transport. CuA, 2 ions that accept electrons from cytochrome C, and then CuB is bonded to a heme A3, forming a binuclear center that transfers 4 electrons to oxygen. So those 4 electrons that ultimately go to oxygen form water, right? And this is complex 4, so we have the intermembrane space, the matrix side, heme A here, and then heme A3 there. And then this, what is that? Is that oxygen? So cytochrome oxidase passes those electrons to oxygen. 4 electrons are used to reduce 1 oxygen molecule into 2 water molecules. 4 protons are picked up from the matrix in this process. And then 4 additional protons are passed from the matrix to the inner membrane space. So again, that the water itself picking, or the oxygen itself picking up those protons is lowering the concentration of protons in the matrix, and then you pump 4 more protons in the matrix. So you're really keeping the concentration of protons low in the matrix, and that all goes to increase that proton motive force that will ultimately produce the ATP, right? And this is the electron flow through complex 4. So cytochrome C brings the electrons to the subunit CuA. Those transfer it to heme A. This is the iron sulfur center. To CuB. In the process, we get 4 protons. Those electrons go to oxygen. and produce 2 waters, and at the same time we pump 4 protons to that inner membrane space. So we get multiple complexes that associate together, and when they do this, they're called the respiratory zone. So, and they're all close together, so you get that quick transfer of electrons, and it's quite efficient that way. And you can actually see them. This is what kind of image is this? I think it's an electron microscope. Yeah, this is from yeast, complexes 3 and 4 together. From yeast, visualized by the electron microscopy after staining with uranyl acetate. And this is the electron microscope image, and then this is where you overlay the crystal structure of complexes 3 and 4 together. So complex 4 is green, and then complex 3 is the red structure. So cytochrome C doesn't have far to diffuse to going from 3 to 4, right? So, and this is basically the summary of the electron flow. So NADH donates its electrons to complex 1. Those go to ubiquinone to form ubiquinol, and we get 4 hydrogen ions pumped into the inner membrane space. That ubiquinol will diffuse to complex 3, where you'll get 4 more hydrogen ions, and then we'll reduce 2 cytochrome Cs from that, which will go to complex 4, where it'll form, half, 1 water molecule from, half an oxygen molecule, because 2 electrons in that process. Whereas complex 2 will take electrons from succinate in the form of FADH2 and reduce ubiquinone to ubiquinol, which will then do the same thing, go to complex 3, reduce 2 cytochrome Cs, which will ultimately, the 2 together will form 2 water molecules, and we get all those protons pumped across. Now again, I'm not going to make you count protons, but just understand how we get that gradient and what that gradient means. But this is the overall summary. So from complex 1 to complex 4, from 1 NADH, we get 10 protons pumped across and 1 water molecule made. From complex 2 to complex 4, we get 6 protons pumped across, an FAD, and then 1 water molecule also. So we get more energy coming from the NADH rather than from FADH2. Right? So just understand that difference. The difference in the number of protons transported reflects the differences in the amount of ATP you can synthesize with either NADH or FADH2. And if you remember, it's that complex 1 versus complex 2, that's the big difference. Complex 1, we pump protons across. Complex 2, we do not, right? So reactive oxygen species can damage the biological macromolecules. So if you let these get out of whack, you can generate oxygen radicals. So, but there are enzymes that will, come and quench those. If we generate a lot of oxygen radicals, we can end up with a lot of oxidative damage. It'll damage these complexes and ultimately result in energy dysfunction in the cell. But luckily we have things that can strip these electrons from the radicals. So we have superoxide dismutase that can take those electrons, convert it to hydrogen peroxide, which will then, glutathione peroxidase will strip the electrons from the hydrogen peroxide and produce water again, converting glutathione, reduced glutathione, take glutathione reductase and reduce that, ultimately transferring them back to NAD. So you have reduced glutathione, they pick up the electrons to form the oxidized glutathione, and the SS means there's a disulfide bond there. And in glutathione reductase, those electrons ultimately get transferred to this will be re-reduced back to the reduced glutathione with glutathione reductase. And then the glutathione protein thiol reduction looks like this. You have a disulfide bond. It'll take the GSH, the reduced glutathione. These electrons will then be transferred here to split this to the active from the inactive, and that's a sign of oxidative stress. It'll go back to this direction. So now we've generated all those protons, which is what causes this proton motive force. The proteins in the electron transport chain create the electrochemical proton gradient, and they do that by actively transporting protons across the membrane, complex 1 and 4, chemically removing protons from the matrix, and that's the reduction of coenzyme Q, and the reduction of oxygen takes protons from the matrix to make that alcohol and water. And then it releases protons into the inner membrane space with the oxidation of, ubiquinol, or ubiquinol. So that has this net effect of producing lots of protons on the inner membrane space, and then you have a lot of hydroxide, or less protons. A lot of negative charge on the inside, positive charge on the outside. It really strengthens that electrochemical gradient. Protons want to flow down their electrochemical gradient, not just because of their concentration, but because of the charge difference across that membrane. And the electron transport is what sets up this proton motive force. The energy of proton motive force drives the synthesis of ATP. So we get the flow of protons down through the ATP synthase, and that causes conformational changes in this F1 fragment or subunit that will force ADP and phosphate to form ATP, using the energy from that electron motive force. Yeah?
Is there a strict ratio of how many protons come back through that ATP synthase to make ATP? It just says
in. Yeah, there is a.
That number, or is it kind of?
There is a number, but it's all going to be dependent on the gradient you have across here, right? Whether how strongly each proton will be able to turn, because this actually rotates. Similar to how you see flagella rotate, but instead those rotate by pumping protons. Those rotate by burning ATP. So you could run this in reverse and pump protons if you want to. But this is all dependent on the gradient across and how much energy you're going to get from going from this side to this side.
Also, so the stronger the gradient,
the more energy each proton will provide, right?
So it'll take less to make each.
Right. So, electron transport is coupled to ATP synthesis. And as described, ATP synthesis requires electron transport, but electron transport also requires ATP synthesis. And here, you can uncouple the two. Have you ever heard of that, uncoupling? So, I'll show you uncoupling in a minute. But the mitochondrial ATP synthase complex contains two functional units, your F1, which is a soluble complex in the matrix, and it individually catalyzes the hydrolysis of ATP. F0 is the integral membrane complex, transports the protons from the inner membrane space to the matrix, which dissipates that proton gradient. The energy transferred to F1 then catalyzes the phosphorylation of ADP. So this F1 unit will hydrolyze ATP if you don't have that gradient forcing it to produce the phosphorylation of ADP to ATP. And the F1, which catalyzes the ADP plus phosphate to ATP, is a hexamer arranged in 3 alpha beta dimers. Dimers can exist in 3 different conformations. So you want to know these 3 conformations and the order at which they change. So you have an open conformation which doesn't bind anything. It's just open. It doesn't have enough, has a low affinity for ATP and a low affinity for ADP and phosphate. A loose conformation will bind ADP and phosphate. And then you have a tight conformation which binds ATP. It catalyzes the ATP formation and binds the product. So it has a high affinity for ATP. So as you switch from one conformation to the other, that's how you get that cycle. The proton gradient causes these subunits to have these conformational changes as it rotates. So you start off with an open conformation and you have nothing bound. You get one turn and it switches to the loose conformation where it will bind ADP and free phosphate. Okay? Then it switches to the tight conformation, and in the tight conformation it really would rather bind ATP. It has a high affinity for ATP, so it catalyzes the ATP formation from the phosphate and the ADP. And then it switches conformation again to the open conformation, and then that ATP no longer has an affinity for the ATP synthase, so it'll diffuse, it'll disassociate from the ATP synthase, and then that's how you form the ATP. And they show it here. So you want to know this, and it's called the binding change mechanism. So the loose conformation will bind ADP and phosphate. It'll switch then to the tight conformation where it forces the production of ATP from the ADP and phosphate. Then it switches to the open conformation where ATP will diffuse away, and then it'll switch to the loose conformation again. And it repeats that as the protons force conformational changes flowing down their gradient. So coupling proton translocation to ATP synthesis. The proton translocation causes a rotation of the F0 subunit and the central shaft gamma. This causes the conformational change within all the alpha-beta pairs. So as you rotate that shaft, it causes that conformation in those alpha-beta pairs on the F1 subunit. Conformational change in one of 3 pairs promotes the condensation of ADP and phosphate into ATP. So, and here's where you get the evidence of rotation. They in this experiment, what did they do? So they have these they added histidines to the different subunits, so it will, and then they have a nickel complex on a slide, so it stays in one particular location on the slide. You have biatenylated actin filament, and then they can rotate that. Let's see. So you have the biatenylated, so the protein avidin binds to biotin very tightly. Have you heard of that? So you have avidin bound to the actin filament. This is biatenylated, the C10. And then that actin filament's got a fluorescent probe, so it kind of amplifies the rotation signal that you'll see. So you have a fluorescent probe here, and you can visually watch it as it spins to get that rotation. So that's.
What would the time on the rotation on that thing is?
It's got to be fast, right? Don't you think they might say? The filament rotates, it makes discrete jumps about every 11th frame, but they don't tell you the frame rate. Oh, here, the position of the actin filament at intervals of 133 milliseconds. So each image is 133 milliseconds apart. So transport of ADP and phosphate into the matrix. So in order to make the ATP, you have to have both phosphate and ADP there, right? But that poses a problem, doesn't it? Because the matrix has a high negative charge compared to the positive charge of the inner membrane space. ADP has 3 negative charges that has to come in, and then phosphate, phosphoric acid, also has a negative charge that has to come in. So you're driving more negative charge into an area with a lot of negative charge already. However, the ATP can translocate out. You're taking those 4 negative charges back out along with it. And then we've talked about this before, right? The malate aspartate shuttle, which basically has the net effect of taking NADH from outside to inside the mitochondria. Or no, this inner membrane space to the matrix, yeah. So we start off with NADH, because NADH won't be able to cross the inner membrane. NADH will reduce oxaloacetate to malate. Then there's a malate alpha-ketoglutarate transporter that can carry the malate across, where NAD will pick up the electrons from malate again and produce oxaloacetate again. And then oxaloacetate via aspartate aminotransferase will be converted to aspartate, and then aspartate can then be transported back out, where it will again be catalyzed into glutamate from alpha-ketoglutarate back to oxaloacetate via aspartate aminotransferase. And it's a cycle just to get electrons from glycolysis and other processes, right, that happen in the cytosol into the matrix. And then there's the glycerol 3-phosphate shuttle, which is another way to get those electrons. So with glycolysis, glycerol 3-phosphate dehydrogenase can take the electrons and dihydroxyacetone phosphate to form glycerol 3-phosphate. Glycerol 3-phosphate then will transfer those electrons to FAD to form FADH2, which will transform them directly to ubiquinol and go to complex 3. So with this, though, you don't get that extra added, protons pumped across, because you're taking from NADH and then going directly to ubiquinone to complex 3. So you skip complex 1 like you would normally with the NADH electrons. So this is your ATP yield from complete oxidation of glucose, and it's going to be 30 or 32. You probably had to answer that on a core bio or microbio test before, so I'm not going to ask you how many electrons are yielded via glycolysis. But the regulation of oxidative phosphorylation is primarily regulated by the availability of substrates, either NADH or ADP and phosphate. Due to the coupling of both substrates required for electron transport and ATP synthesis. You need the ADP and phosphate for the synthesis of ATP, NADH you need for the transport of electrons. Inhibitor of the F1 or IF1 prevents hydrolysis of ATP during low oxygen. If you don't have oxygen available, you don't want to perform oxidative, reactions. It can cause those radicals that you can generate. Only active at lower pH, encountered when electron transport is stalled. Electron transport stalls when you have low oxygen. Inhibition of oxidative phosphorylation will lead to the accumulation of NADH, causing feedback inhibition back up to, do you guys remember this?
Phosphofructokinase 1?
Yes, phosphofructokinase 1, which is the entry step into glycolysis, right? The committed step. And this is the overview of that regulation, right? So phosphofructo, there's glucose 6-phosphate, and then phosphofructokinase 1, activated by AMP and ADP, inhibited by ATP and citrate. Whereas NADH activates that respiratory chain, and ADP informs NAD. So if you lack NADH or ADP and phosphate, the regulatory chain will, your respiratory chain won't be active, and you'll build up, the ADP and phosphate, which will then activate it. So light energy is converted to ATP in the plant chloroplast, and it's a similar type of process, but rather than getting the electrons directly from NAD from burning sugar or fats, we capture those electrons from water using light as the energy source. And it's broken up into two different phases. You have the light reactions and then the dark reactions, aka the carbon assimilation reactions. So in the light reactions, we produce NADPH and ATP. In the dark reactions, we use both of these to produce sugar from carbon dioxide. And you have various pigments that are going to be able to absorb different wavelengths of light energy. There's chlorophyll A, and if you look, it has a long phytol tail, which means it's going to be lipid soluble. Instead of iron being coordinated in the center, it uses magnesium. Then you have other pigments such as beta carotene. We've heard of this, right? You get this from carrots, and it gives you that orange color. Lutein, there's xanthophylla, or phycotherapy. I'm not good at saying words. Photopigments are going to absorb different wavelengths of light. Chlorophyll B absorbs here. Beta carotene over that period. Then we have phycocyanin and phycourethrin, different wavelengths. And then chlorophyll A is way over here and here. So, and then this is the basically the wavelength of visible light coming from the sun. So you get a whole range of pigments that absorb over a whole range of wavelengths, and that's just to capture the most energy as possible. And then there's a sort of an organization for the light absorbing molecules in chloroplast. So you'll have these antenna chlorophylls that can absorb the light, and they'll feed that energy via electron transfer to a reaction center, and that reaction center is where you get converts the energy from that photon into separation of charge, which is an oxidation reduction reaction, right? And that initiates that electron flow. So from light energy to charge separation, so we get light, a photon of light boosts the electron at an antenna molecule. It transfers that energy to a neighboring molecule, which will boost it, which will then transfer it. Ultimately, it transfers from antennae to antennae to antennae. And we're talking about the energy, not the electrons. Okay. Ultimately to the electron acceptor at the antennae. And then that electron acceptor at the antennae, the excited reaction center chlorophyll, passes an electron to an electron acceptor. The electron hole in the reaction center is then filled by an electron from an electron donor. The absorption of a photon then basically has charge, the net separation of charge. That electron donor will ultimately be water, right? So we get water donating an electron, and that will evolve half of an oxygen molecule. That electron gets boosted here to P680, to the P680 star, which is the excited state, and then that electron will flow down its reduction potential to different subunits. In the process, it goes to cytochrome B6F complex, which generates a proton gradient. It goes to the P700 complex that can absorb another photon and light to boost it here. Electron goes down its gradient again, which ultimately NADP will accept that electron to form NADPH, which this NADPH can then be used for anabolic processes. And the anabolic processes that use it during the dark reactions is the synthesis of carbohydrates from carbon dioxide or carbon fixations. It's very similar to the electron transport chain. So photosystem 2 involves oxygen, and this happens during the light reactions. Water comes in, the electrons are stripped from the water, and ultimately generate the oxygen. And this shows you the reactions that occur for the water splitting. First exciton. So here you have water bound. Electron comes from that to the first exciton, and then the second. We lose 2 protons in the process. The third exciton, and then the fourth, and then ultimately you get oxygen released from that process. And then photosystem 1 results in the reduced NADPH from NAD. So you get exciton transfer of electrons that ultimately end up with the ferredoxin, and you'll end up with the NADH. And analogous to the electron transport that we see for a mobile electron transporter, we have, instead of ubiquinone, it's plastoquinone that accepts electrons, and you have a very similar Q cycle that we would see in the electron transport chain. And this is the organization of the photosynthetic machinery in the alkaloid membrane, and so forth. So I'll tell you, I'll be focusing more on electron transport than photosynthesis, but just understand the difference between the dark and the light cycles. And then that's basically all I was going to be covering in chapter 19.